Hello everyone, myself Dr. Amanpreet Kaur Kalsi. In this lecture, we will take up another topic from developmental biology, which is early development in chick. The topics we will cover in this lecture are fertilization in chick, cleavage, that is the process of blastulation in chick, fate map of chick, and gastrulation in chick up till the formation of 3 germ layers. Let's have an overview of chick life cycle. The adult hen undergoes fertilization and lays egg, which has a small disc of blastoderm lying atop a yolk-laden egg. This egg undergoes the process of cleavage, forming area pellucida, area opaca, and marginal zone. Up till this, the events occur within the oviduct and takes around 20 hours after fertilization. At this time, egg is laid and the process of gastrulation occurs, which gives rise to primitive streak and hensen's node and formation of 3 germ layers. This takes around 16 to 50 hours after laying of egg. Then occurs the process of organogenesis, where organs develop. After this, egg hatches to form chick, which grows to give rise to adult. In this lecture, we will take up early development in chick up till gastrulation, that means the formation of 3 germ layers. Let's start with fertilization. Fertilization in hen occurs in the oviduct, which is shown here in grey part. This shows the enlarged view of oviduct. This is the point where fertilization occurs. After fertilization, albumin, the white colored one, and shell are secreted within the oviduct to cover the chick egg. After fertilization is the process of cleavage. Cleavage in chick depends upon the kind of egg they have. Chicks have telolecithal eggs where large amount of yolk is present in the egg and only a small disc of cytoplasm is available at the animal pole. Because of this, the cleavage in chick is discoidal meroblastic. Discoidal means it occurs in a disc of cytoplasm which is available. This disc of cytoplasm is called blastodisc or germinal disc. It is a small disc of cytoplasm 2 to 3 millimeters in diameter at the animal pole of egg cell. And meroblastic cleavage means the cleavage is incomplete, occurring only in the blastodisc and not extending throughout the egg. If you see a real egg, this is how germinal disc will appear in the yolk. Let's take up the process of cleavage step by step. This figure shows the discoidal meroblastic cleavage as it occurs in chick, 4 stages viewed from the animal pole. The first cleavage furrow appears centrally in the blastodisc or germinal disc. Cleavages do not extend into the yolky cytoplasm, so they occur only superficially, and because of this, the early cleavage cells are continuous to one another and have large amount of yolk underside them. This results in the formation of single-layered blastoderm. As shown in the E part of the figure, where an early cleavage embryo is shown from the side, that is mid-sagittal section. As further divisions occur, the equatorial and vertical cleavages divide the blastoderm into 5 to 6 cell layer thick tissue. After this, the deep cells in the center of blastoderm are shed and die, giving rise to a single cell layer center which is called area pellucida. The area surrounding the area pellucida, where the cells are not shed and are still multi-layered are called area opaca. And the zone between area pellucida and area opaca is called the marginal zone. As cleavage proceeds, the blastoderm cells absorb water from albumin and secrete fluid between themselves and the yolk, giving rise to the cavity called subgerminal cavity. Next comes the fate map of chick. This figure shows the fate map of chick at early gastrula stage, the dorsal surface view, looking down on the embryo at what will become its back in future. A indicates the anterior of the embryo and P indicates the posterior end of the embryo. Cells that will form notochord, the dark red colored ones, occupy a central dorsal position, while the precursors of neural system, shown in dark blue color, lie immediately anterior to notochord precursors. Neural ectoderm is surrounded by a less dorsal surface ectoderm, which will form the epidermis of skin. The other mesodermal derivatives are shown in red color, dark red and light red ones, and the cells which will form endodermal parts in future are shown in yellow color. The dashed green line signifies the site of ingression. That means the path cells will follow as they migrate from the exterior to the interior of the embryo. After fate maps, let's come to gastrulation. Gastrulation in chick is a combination of different processes which are hypoblast formation, primitive streak formation, migration through primitive streak, regression of primitive streak, and finally epiboly of ectoderm. We will take up each one of them one by one. By the time a hen has laid an egg, the blastoderm contains some 20,000 cells. At this time, most of the cells of area pellucida remain at the surface, forming the epiblast, shown in the figure with grey colored cells. Some area pellucida cells migrate individually into the subgerminal cavity, giving rise to polyinvagination islands of 5 to 20 cells each. These cells form the primary hypoblast. Shortly thereafter, sheet of cells from the posterior margin migrates anteriorly, pushing the primary hypoblast cells towards the anterior side. These cells form the secondary hypoblast. The cells of posterior margin which migrate anteriorly comes from the collar sickle region and are distinguished from rest of the cells of posterior margin. Collar sickle is a local thickening in the posterior margin. The resulting two-layered blastoderm, that means the epiblast and hypoblast, is joined together at the marginal zone of area opaca, and the space between the layers forms the blastocele. The epiblast gives rise to all the three germ layers of embryo proper and a considerable amount of extraembryonic membranes, while the hypoblast gives rise to portions of external membranes, including yolk sac and stalk, primordial germ cells, and it also provides chemical signals which specify migration of epiblast cells. After hypoblast formation, the next main step is the formation of primitive streak. Primitive streak is the major structural characteristic of avian, reptilian, and mammalian gastrulation. Primitive streak cells arise within posterior marginal zone, shown in the figure in red color. The first visible as cells accumulate in the middle layer between epiblast and hypoblast. The epiblast at posterior marginal zone becomes thick just anterior to the collar's sickle, shown here in purple color. These figures shows entire embryo viewed from the ventral side in B and from the dorsal side in figure C. The red color one shows the primitive streak as it's seen from the ventral side of the embryo and the dorsal side of the embryo. The blue colored ones over here are the cells of secondary hypoblast and the green one are the cells of primary hypoblast. As primitive streak elongates, as we will see later on, the secondary hypoblast cells also moves anteriorly and in some way guides the movement of primitive streak. This figure shows the dorsal view of formation of primitive streak. Blastoderm here is seen at 3 to 4 hours after fertilization. The thickening of primitive streak is initiated by an increase in height of cells forming the center of primitive streak. Then the surrounding presumptive streak cells digest the underlying extracellular matrix and become globular and motile. This results in intercalation between these cells and convergent extension, giving rise to progression of primitive streak. Thus, the primitive streak undergoes convergent extension, converging from sides and extending anteriorly. This figure shows the blastoderm at 7 to 8 hours after fertilization. The doubling in streak length is accompanied by a concomitant halving of its width. This figure shows the formation and elongation of primitive streak at 15 to 16 hours after fertilization. As the cells converge to form primitive streak, a depression forms within the streak. This depression is called primitive groove, and it serves as an opening through which migrating cells pass into the blastocele. Thus, the primitive groove is homologous to the amphibian blastopore. At the anterior end of primitive streak is a regional thickening of cells called the primitive knot or hensen's node. The center of this node contains a funnel-shaped depression, sometimes called the primitive pit, through which cells can pass into the blastocele. Hensen's node is the functional equivalent of dorsal lip of the amphibian blastopore. The streak eventually extends to 60 to 70% of the length of area pellucida. The next step is migration through the primitive streak. Along with lengthening of primitive streak, migration of cells start through the streak. The second figure here shows fate map of chick epiblast for definitive primitive streak stage. The movement of mesodermal precursors through the primitive streak is shown here. Notochord precursors enter the embryo through the hensen's node. The somites precursors enter through the anterior part of primitive streak. From the middle region of primitive streak, lateral plate mesoderm cells enter, and from the posterior region of primitive streak, the cells which will form extraembryonic mesoderm enters the embryo. The sequence in which cells migrate through the hensen's node is as follows. The cells to enter first are the pharyngeal endoderm of foregut. The next cells to enter from the head mesenchyme and precaudal plate mesoderm. After this, the third one to enter through the hensen's node are cauda mesoderm cells, which form head process and notochord, and then are the cells which form notochord, which enter through the hensen's node. This figure also shows migration through the primitive streak. A stereogram of gastrulation chick embryo showing the relationship of the primitive streak migrating cells and the hypoblast and epiblast of the blastoderm. Cells migrating through the hensen's node travel anteriorly to form the precaudal plate and notochord. Those migrating through the next anterior region of the streak travel laterally but converge near the midline to make notochord and somites. Those from the middle of the streak form intermediate mesoderm and lateral plate mesoderm. Meanwhile, cells continue migrating inwardly through the lateral and posterior portions of primitive streak. As they enter the blastocele, these cells separate into two layers. The deep layer joins the hypoblast, as shown here in yellow-colored cells, along its midline and displaces the hypoblast cells to the sides. These deep-moving cells give rise to all the endodermal organs of the embryo, as well as to most of the extraembryonic membranes. The second migrating layer, shown in pink color in this figure, spreads between the endoderm and epiblast, forming a loose layer of cells. This middle layer of cells generates the mesodermal portions of the embryo and mesoderm lining of extraembryonic membranes. The hypoblast cells eventually sort out to form a layer beneath the endoderm and contribute to the yolk sac. By 22 hours of incubation, most of the presumptive endodermal cells are in the interior of the embryo, although presumptive mesodermal cells continue to migrate inward for a longer time. Now, a new phase of gastrulation begins. While mesodermal ingression continues, the primitive streak starts to regress, moving hensen's node from near the center of area pellucida to a more posterior position. As you can compare over here in the figure, which shows formation of notochord and mesodermal somites as primitive streak regresses at 19 to 22 hours, 23 to 24 hours stage after fertilization, and F figure showing 4 somite stage. The hensen's node, which was almost in the center of area pellucida at this stage, gradually is regressing towards the posterior end and has reached the posterior end in this figure. The regressing streak leaves in its wake the posterior dorsal axis of the embryo, including the notochord. As hensen's node move caudally, the posterior region of notochord is let down. Finally, hensen's node regresses to its posterior position, forming the anal region. This figure shows the regression of primitive streak, leaving the notochord in its wake. Various points of the streak are represented by letters D, B, C, were followed after it achieved its maximum length. The x-axis shows hours after achieving maximum length. After achieving maximum length, the primitive streak takes about 20 hours to regress. This is the path which it follows in regressing, and in its wake of regression, it leaves behind the notochord. Therefore, as primitive streak regresses, the notochord keeps on elongating. Next is epiboly of ectoderm. While the presumptive mesodermal and endodermal cells are moving inward, the ectodermal precursors proliferate and migrate to surround the yolk by epiboly. The enclosure of yolk by ectoderm is a big task and takes the greater part of 4 days to complete. It involves the continuous production of new cellular material and migration of presumptive ectodermal cells along the underside of vitelline envelope. As avian gastrulation comes to a close, the ectoderm has surrounded the yolk. This ectoderm gives rise to epidermis and skin. Endoderm has replaced hypoblast. This endoderm gives rise to musculoskeletal, cardiovascular, and urogenital systems, and mesoderm has positioned itself between these two regions. Mesoderm ultimately gives rise to the lining of gastrointestinal and respiratory tract. This is all about early embryonic development in chick. Major parts of this lecture were taken from the book Developmental Biology by Scott F. Gilbert. Thank you so much.